Can I have everyone return to their seats, please? Hi, everyone. I hope you enjoyed the lunch. We are going to start back up again. I think there's a few more people trickling in. I'd like to go ahead and introduce Dr. Resmi Ravindran. Yeah. She's from the University of California, Davis. And I've known Resmi now for probably 10 to 12 years. Yeah. So it's a pleasure for me to introduce her to you all. Resmi is currently working as a senior scientist in a clinical proteomics core lab in the Department of Pathology at the University of California, Davis Medical Center. She has a broad background in infectious pathogen immunology with specific training and expertise in developing highly efficient and high throughput multiplex approaches for disease-related immune biomarker studies. Dr. Ravindran is a key researcher in several collaborative and national and international infectious disease research with scientists and clinicians in the US, India, Uganda, and Pakistan for the chronic lung infection disease, tuberculosis. With that to do, I'd like to introduce you all to Dr. Ravindran.
Thanks, Cassie. Good afternoon, everyone. I hope all of you had a nice lunch. So today I will be talking about the evaluation of a bead-based multiplex SARS-CoV-2 variant neutralization antibody assay for vaccine efficacy assessment. So here is the agenda for today. I'll discuss the various SARS-CoV-2 neutralizing antibody tests currently being used and introduce a bead-based multiplex viral neutralization test from Bio-Rad Laboratories and its comparable performance with the gold standard neutralization test, which is the whole or live virus neutralization test in COVID-19 patients. Next, I will talk about the evaluation of the bead-based multiplex viral neutralization test in individuals immunized with Pfizer and Moderna vaccines, and how binding antibodies correlate with neutralizing antibodies in this group. I'll also talk about the evaluation of the bead-based multiplex viral neutralization test in animal models for COVID-19. Finally, I'll highlight the benefits of the bead-based multiplex viral neutralization test. As we all know, when SARS-CoV-2 infects the host cell, the receptor binding domain, abbreviated as RBD, on the spike protein on the surface of the virus, interacts with ACE2 receptors on the host cell surface. This interaction helps internalization of the virus, leading to its replication and subsequent infection. When the virus infects the host cell, antibodies are generated against various regions of the virus. These antibodies are broadly classified as binding antibodies or neutralizing antibodies. Binding antibodies are able to bind to the virus, but are not able to prevent the interaction between the RBD and the ACE2 receptor, whereas neutralizing antibodies, as the name suggests, are able to neutralize the virus. That means they are able to bind to the receptor binding domain on the spike protein, thus preventing its interaction with the ACE2 receptor. This in turn prevents internalization of the virus and its subsequent infection. So various commercial assays are currently available. They either look at the binding antibodies or neutralizing antibodies. Binding antibodies provide the information whether the individual is infected or not, whereas neutralizing antibodies provide the information whether the individual is protected from the infection or not. A robust SARS-CoV-2 neutralizing antibody test to look at the neutralizing antibodies against SARS-CoV-2 is needed, not only to evaluate the vaccine efficacy in clinical trials and after large-scale vaccinations, but also for convalescent plasma screening and to identify who needs a booster shot. FDA has approved several neutralizing antibodies for treatment of COVID-19. So I'll talk about the various neutralizing antibody tests currently being used. Gold standard is the whole virus or live virus neutralization test, abbreviated as WVNT. Here, live SARS-CoV-2 virus is used to infect host cells expressing ACE2 receptors. This is followed by the addition of patient plasma or serum containing neutralizing antibodies, and neutralizing capacity is assessed by looking at the formation of viral plaques. Since this test uses live SARS-CoV-2 virus, the test needs to be performed in a biosafety level 3 laboratory by trained personnel. Hence, this is expensive and takes 3 to 5 days to get the final results, so the turnaround time is really slow. A variation of the WVNT is the pseudovirus neutralization test. Here, instead of live SARS-CoV-2 virus, a pseudovirus, for example, a lentivirus expressing SARS-CoV-2 spike protein is used to infect the host cell expressing ACE2 receptor, and the test is performed as above. So this also, even though this doesn't use live SARS-CoV-2 virus, this still uses live virus. So the test needs to be performed in a biosafety level 2 laboratory by trained personnel, and the turnaround time is similar to WVNT. So both these tests are cell culture based, hence reproducibility is limited and not amenable to high throughput. A new addition to the neutralizing antibody test platform is the bead-based neutralization antibody test from Bio-Rad Laboratory. Here, instead of live cells and cell culture, magnetic microbeads are used. So I'll explain the principle of this test in my next slide. Schematic here shows how the bead-based neutralization antibody test from Bio-Rad Laboratory works. So this is a Luminex-based competitive assay. Magnetic microbeads are used here. And proteins from SARS-CoV-2 virus, either the spike protein or receptor binding domain protein, is covalently coupled to magnetic beads. Beads are incubated with patient plasma or serum sample containing neutralizing antibodies. This is followed by the addition of biotinylated ACE2 receptor. So I had mentioned in my previous slide that neutralizing antibodies are able to bind to the receptor binding domain on the spike protein. So this binding interaction is utilized to develop this test. So neutralizing antibodies in the patient plasma or serum compete with the biotinylated ACE2 receptor to bind to the SARS-CoV-2 proteins on the bead surface. And streptavidin 5-coerythrin is used as the reporter, and output is the median fluorescence intensity. So higher the neutralizing antibodies in a sample, lower the binding of biotinylated ACE2 to the bead surface. That means higher the neutralizing antibodies in a sample, lower the median fluorescence intensity. Final results are expressed as percentage inhibition with respect to the negative control sample. So since this test uses a multiplexing approach, there is flexibility to add beads coupled to SARS-CoV-2 proteins from several variants of the virus in the same reaction. So we have collaborated with Bio-Rad laboratories to evaluate the utility of the Bioplex VNT in our studies. For the Bioplex VNT, plasma samples were serially diluted to determine the optimal dilution that gave the highest correlation between the Bioplex viral neutralization test and the gold standard test, which is the whole virus neutralization test. Plasma samples from 4 COVID-19 patients were collected over several days post-symptoms, were selected to perform this experiment, and the samples were serially diluted, as I mentioned. Each colored line represents a time point post-symptom, and different serial dilutions are shown here. Optimal dilution was determined as the dilution that gave the highest correlation between both Bioplex viral neutralization test and the whole virus neutralization test. And another criteria to choose the dilution was we made sure plasma samples or similar plasma samples would be predicted to fall within the linear range of the assay and not report as saturated percentage inhibition. A plasma dilution of 1 to 37.5 was chosen as the optimal dilution for Bioplex viral neutralization test, and all subsequent analysis of the study samples were performed at this dilution. Graphs here show the correlation between the Bioplex VNT and the gold standard whole virus neutralization test in the same 4 COVID-19 patients that I had shown in my previous slide. And as you can see, very strong correlations were observed between both tests. We then evaluated the efficacy of mRNA vaccines in individuals immunized with Pfizer and Moderna vaccines using the Bioplex VNT. Heat map here shows the efficacy in individuals who received Pfizer vaccine. And here is the efficacy in the Moderna group. So we looked at neutralizing antibodies against 2 proteins from wild-type SARS-CoV-2 virus, spike-1 and receptor binding domain, as well as spike or RBD of several variants of SARS-CoV-2 virus using the Bioplex VNT. And plasma samples were collected before vaccination, which is a week 0. 3 weeks after the first dose in the Pfizer group, 4 weeks after the first dose in the Moderna group, 4 and 26 weeks after the second dose from both groups. As you can see, the green segment is the baseline, which is week 0 or pre-vaccination time point. None of the study subjects in our group had any baseline reactivity against any of the SARS-CoV-2 proteins tested. Neutralizing antibodies were detected at about 1 month after the first dose in both groups, and the levels of neutralizing antibodies in the Moderna group was about 2 to 3-fold higher than in the Pfizer group for both wild type as well as all the variants. And neutralizing antibodies peaked at 4 weeks after the second dose in both groups, and waning of the neutralizing antibody response was observed in both groups at 26 weeks, which is about 6 to 7 months after the second dose, and the waning was more in the Pfizer group than in the Moderna group. We had developed an in-house assay to detect binding antibodies against SARS-CoV-2 SRBD. So we looked at the binding antibodies such as IgG, IgM, and various IgG subtypes, IgG 1, 2, 3, and 4 in the vaccine cohort. So graphs here show the binding antibodies in both Pfizer and Moderna groups. So this is the total IgG level in both groups, and the levels were similar to what you have seen in the heat map for the neutralizing antibodies. So this is the baseline total IgG in the Pfizer group and Moderna group. And total IgG levels increased at 3 to 4 weeks after the first dose in both groups, peaked at 4 weeks after the second dose. and declined at 26 weeks after the second dose. So even though the total IgG levels declined, they still remained way higher than the baseline in both groups. And IgM was undetectable in both groups after post-vaccination. IgG1 subclass antibodies were very high in both groups. And as you can see, they followed similar pattern as the total IgG levels. And in the Moderna group, we have seen for total IgG as well as various IgG subclass, the levels were much higher than in the Pfizer group. IgG2 levels were high only 4 weeks after the second dose in both groups. IgG3 levels followed similar pattern as the total IgG and IgG1 levels, even though the levels were lower. IgG4 levels were inconsistent in both groups. So graphs here show the correlation between the total IgG and various subclasses and the neutralizing antibodies. So this is the Pfizer group. And here are the correlations in the Moderna group. Correlations between the total IgG and neutralizing antibodies were moderate or medium in both Pfizer and Moderna group at all time points post-vaccination. However, the correlation between the various IgG subclasses and the neutralizing antibodies were very low in both groups. So in parallel to the human experiments, we have evaluated the Bioplex VNT using an elaborate panel of animal sera obtained from various vaccination and infection experiments. Graph here show the correlation. For some reason, it got skewed, sorry. So graphs here show the correlation between the Bioplex VNT and the gold standard whole virus neutralization test in non-human primates immunized with an mRNA vaccine encoding spike protein or adjuvanted spike protein vaccine alone. And I don't think you can see the R-square values here, but very strong correlation was obtained between both tests in non-human primates. Heat map here shows the neutralizing antibody levels in non-human primates immunized with an adenoviral vector vaccine expressing spike RBD protein. 9 animals were used in this study group. Samples were collected at week 0, which is the pre-vaccination time point, as well as several weeks post-vaccination. Similar to humans, neutralizing antibodies were detected at 4 weeks after the first dose in macaques, peaked at 4 weeks after the second dose, and remained high at 6 weeks after the second dose. So in addition to week 0, we have included another group of animals. These are non-human primates immunized with an adenoviral vector vaccine expressing nucleocapsid protein. So this group was also used to determine the assay specificity. As expected, none of the animals in this group had any baseline reactivity with any of the SARS-CoV-2 spike or RBD proteins tested. So these are the binding antibody, which is the total IgG levels in the 9 macaques that are shown in this heat map. And among the 9 macaques, 2 of them were models for HIV infection in humans. So these 2 animals were infected with SIV, which is simian immunodeficiency virus, which is the counterpart of HIV in humans. And these 2 animals were under antiretroviral or ART therapy. As you can see, these animals were able to elicit very strong neutralizing antibodies. So this is very promising for HIV patients. So these are the binding antibodies, a total IgG against nucleocapsid protein in the green animals shown at the bottom in the green segment here. And none of these animals had any cross-reactivity against SARS-CoV-2 SRBD protein. So in addition to macaques, we have evaluated the Bioplex VNT in small animal models for SARS-CoV-2 infections, such as hamsters, rabbits, and guinea pigs. So graph here show the correlation in hamsters between the Bioplex VNT and the whole virus neutralization test. And here is the correlation in rabbits. Very high correlation was obtained between both assays. Heat map here shows the neutralizing antibodies in hamsters experimentally infected with wild-type SARS-CoV-2. So we have used two groups of hamsters who received low dose of SARS-CoV-2 as well as high dose of SARS-CoV-2. Hamsters are highly susceptible to SARS-CoV-2 infection and develop severe disease few days after inoculation. And as you can see, SARS-CoV-2 infection elicited very strong neutralizing antibodies in hamsters, irrespective of the dose used. So this segment shows the neutralizing antibodies in rabbits infected with SARS-CoV. Green is the baseline, which is uninfected rabbits, and the bottom red segment is the neutralizing antibody levels in guinea pig infected with SARS-CoV. So unfortunately, we only had one sample from guinea pig available to test. And as I mentioned, both these rabbits and guinea pigs were infected with SARS-CoV and not SARS-CoV-2. This is not surprising because the spike proteins of both SARS-CoV-2 and SARS-CoV share about 74% amino acid homology, whereas receptor binding domain protein share about 76% amino acid homology. So what you see here is the cross-reactivity between the proteins. To summarize, we have evaluated the Bioplex multiplex neutralization antibody assay using both human and animal sera. In fact, 4 species of animal sera, non-human primates, hamsters, rabbits, and guinea pigs. Very strong correlation was observed between Bioplex viral neutralization test and whole virus neutralization test. Very high sensitivity and specificity was achieved using the Bioplex VNT in human and animal sera. Correlation between the binding and neutralizing antibodies were moderate. So here are the benefits of the Bioplex multiplex neutralizing antibody assay. Unlike the gold standard whole virus neutralization test, Bioplex viral neutralization test does not need to be performed in BSL-3. This makes it widely accessible for both research and clinical applications. Faster and easy to use format enables the handling of large number of samples over a short period of time. It takes only about 3 to 4 hours to get the final results, as opposed to several days using the gold standard whole virus neutralization test. There is an option to run a standard curve and quantitate the levels of neutralizing antibodies in the test. Multiplexing platform enables the detection of 2 wild type as well as 11 variants of SARS-CoV-2 in the same reaction. So this is the panel currently available. Another major advantage is Bioplex VNT is both species and isotype independent. So thanks to all my collaborators, and thank you all for listening.